Today we'll be seeing a liverwort, Reboulia hemispherica, which is otherwise called purple fringed liverwort. It is a thalloid liverwort. It belongs to Hepaticopsida, subclass Marcantiae, Marcantiales, Itoniaceae, Reboulia hemispherica. Hemispherica meaning the notch in the thallus. No, it looks like a hemisphere. Araivattam solvom, hemisphere. So the name comes from the structure of the notch of the thallus. So this is similar to Marcantia in the morphological nature, with some differences, which we'll be seeing in due course. So it normally you find it in Nilgiri Hills, Palani Hills, Tirunelveli Hills. Okay, and it's quite cosmopolitan. You find it throughout the world, and all hilly regions you find this Reboulia species. So this is the gametophytic plant body, thalloid body, again dichotomously branched. This is the notch, the apical cell showing the growth, indefinite growth. Okay, and this is pale green in color, dichotomously branched with undulate margins, so wavy margins you can see, and a distinct midrib. And you can see it's purplish in color. That's why it's called purple fringed liverwort. So this looks like fringes, the undulated margins. And the rhizoids and scales from the ventral side, you can see. And rhizoids are both smooth type as well as pegged rhizoids, we say, tuberculate rhizoids. You see both kinds of rhizoids. And then the scales are also two ranked. Okay, and when the scales are one cell thick normally, they are unappendaged. Unappendaged means they don't have any hair like projections. And a fresh specimen of Reboulia has a distinct aroma. When you crush it, you get a very good aroma, a very good smell. Okay, and this is named after the botanist, Italian botanist Eugen di Riboll. So that is the name given by the Italian botanist. So these are few more pictures of the thallus. Moderate sized thalloid liverwort. Okay, it is similar to Marcantia, but it has some unique structural features. The thallus size is around 6 to 8 millimeters. The width of the thallus, breadth of the thallus is very small, 6 to 8 millimeters. And margins are often purplish. That is why I told you here, you can see the margins are always purplish in color. And mature thalli have purple undersides with 2 rows of purplish scales. Here you can see very clearly with 2 rows of purplish scales. The close-up view of the thallus, ventral surface of the thallus. You can see the scales, 2 rows of purplish scales, and you can see the rhizoids very clearly. You can see the distinct midrib. You can see the purple fringes, the margins which are undulated. So these are unique features of this Reboulia. And you can see whitish pores. You can see white color pores are actually, you can see it with naked eyes, predominantly on the archegoniophore, female receptacle, you call it. No, you can also see it on the thallus, on the dorsal surface of the thallus. You can see whitish pores. Okay, and the margins of the thalli, they curve upward. No, they don't remain flat. They curve upward. That's why they are undulating. Okay, they curve upward. Okay, male noki valayum. And if the small male receptacles you can see are kidney shaped, okay, or crescent shaped. Sessile means they don't have stalk. They are as if they are attached to the thallus. And the male receptacles are located behind each female receptacle on a thallus. That means it's a monoecious thallus. You find both male and female receptacle on the same thallus, and the male receptacles are located 1 to 10 millimeter behind the female receptacle. And you can see white filaments at the apex, at the top portion, as well as the basal portion of the female receptacle. These are protective in function, actually, hair like filaments, white filaments. And the female receptacle has 4 to 7 lobes. So some of them will have 4, some will have 5. So they have seen 4 to 7 lobes. In this, you can see 4 fertile lobes normally at maturity. This is the uh, structural aspect of Reboulia. When you come to the internal structure, it is similar to what we see in Markantia and Rixia. You have a lower colorless region of elongated cells with thick walls and upper region of loose chlorophyllous tissue. Okay, air chamber. You have a lot of air chambers and parenchymatous, thin-walled parenchymatous cells with chlorophyll. Okay, and they have seen that intracellular fungi live in the lower region of the thallus. This is the upper region of the thallus. In the lower region of the thallus, they have seen fungal hyphae. So it has been documented that fungal hyphae live in the lower region of the internal thallus. And here, this is the growing point of the wherever you have the notch, no, this is the notch, we say it. This is the growing point. Okay, growing point. There, the cells are thick walled. Okay, epidermal cells are slightly thickened and devoid of chloroplast. But you see here, you can see the epidermal cells are having chloroplast. Okay, except near the growing point of the thallus. So they are having pores also. The air chambers open to pores. There are pores. The male receptacle and female receptacle are lined by these kinds of pores. Okay, the plastids in the air chamber region are rather large. Okay, wherever you find the chloroplasts in the air chamber region, they are quite large. They contain several starch grains. Okay, and small oil globules are also found abundantly in the cells. Okay, and no gemmae are produced in Reboulia. No gemmae are produced. This is another. It is a major difference between the other Markantiaceae members that we have studied before, Markantia rixia. Here you don't find any gemma production. Now, when you talk about the growth, apical growth, the thallus grows by means of a single cuneate apical cell. Cuneate, a triangular cuneate apical cell. Okay, the thallus grows by means of this apical cell, which cuts off segment from all its 4 sides. So you get a thalloid dorsoventral thallus. And the air chambers form a very extensive system of irregular air passages. So previously, when it's growing, the growing stage, the cells are together, tightly packed. But as the differentiation continues, the some of the cells lose their cell wall. No, there is disintegration of the cell wall at places. So it leads to air cells with a lot of air chambers. Okay, so how does this kind of thallus arise from this kind of thallus? It is due to the disintegration of the side walls of some of the cells. So you get cells with large intercellular spaces. Okay, and you have the air chambers arise immediately behind the apical cells. See, that's what is given here. Immediately behind the apical cell of the thallus by intercellular splitting. Okay, and it goes on till the mid portion of the thallus. And you can see in male receptacle and female receptacle, the pore formed by the the epidermal cells. Epidermal cells, they grow in such a way that they form a pore just above the air chamber, just above the air chamber. See, in a female receptacle, the epidermal cells form the barrel-like pore. They take a shape of a barrel. The pore looks like a barrel shaped. But in a male receptacle and 3D receptacle, it is a simple pore. It is a simple pore. and through this pore only the sperms will get out. Okay, they will get dispersed, they will get released from this uh, simple pore only. Okay, and here you can see a little bit about the what to say, female and the male part of the thallus. The liverbird is monoecious, which means both the male and the female receptacles or the sex organs are produced by the in the same thallus itself. Both the female and male reproductive organs are present on the same plant. The female reproductive organs are called archegonia. They are called the archegonia. They are contained in a shallow dome-shaped receptacle, okay, that develops within the notches of the, these are the notches. These are the notches of the growing tip. So they are developed as dome-shaped receptacles, okay, and the male reproductive organs are located just beneath the female receptacles. Okay, disc shaped, reniform shaped, sessile. Okay, male antheridia are formed just beneath the archegonia. Okay, they are just located behind the female receptacles on the same thallus or the same branch. So after the fertilization, the male receptacles become shriveled, they fall off. But the female receptacle, what happens? They just elevated, they get elevated on a stalk. Okay, they get elevated. So they are actually here near the notch of the thallus. But then after the fertilization, they get elevated. The archegonia 4 or the female receptacle gets elevated on a stalk. Okay, this is the seta. This is the seta. And so depending on the environmental condition, it comes in different colors. Okay, and the dome shaped head of the female receptacle. Okay, the dome, this is the dome shaped head. You have 4 dome shaped head. This is called carpocephalum. This is called carpocephalum. So they become more lobed. Okay, the lobes the lobes are shallow in the beginning, then they become deeper. Okay, you can see on the undersurface of the lobes, the sporangium or the sporophyte develops. Okay, so they have some 4 to 7 lobed. If they have 4 to 7, so 4 to 7, the sporogonium, which you call it, sporogonium develops on the undersurface of the archegonium. So there is a ring of spore cases. You have a white membranous covering which is surrounding the sporogonium. Okay, and you have white filaments also. I told you they are protective against the developing sporogonium. And once they are matured, the spore case or the spore wall breaks, gets disintegrated, and they release the spores to the wind actually, and they don't have uh, a lot of elaters. You have seen there are little bit, I mean, you don't find innumerable elaters here in Reboulia. These are some pictures of the female receptacles. The female receptacles, once they matured, they raise to the stalk. You can see the stalk, you can see the white filaments, okay, very clearly. This is the thallus with the purple fringe. You can see the purple fringes very clearly in this picture. Yeah, this is a single archegonia 4 with the developed sporogonium. You can see the white membrane very clearly. So as it matures, it will get smaller and smaller, and the sporogonium will liberate the spores. Yeah, in this picture also you can see it very clearly. This is the diagrammatic representation of the thallus with the, this is the antheridia, okay, and the archegonia, female receptacle and the male receptacle. Male receptacle is sessile, female receptacle is stalked. Okay, this is the internal structure and the pore. I told you, you see white pores are found very commonly on the dorsal surface. These are the pores. These are the ventral scales. You can see the midrib. These are the ventral scales, and you have the rhizoids. Yeah, this is another picture of, you know, uh, Reboulia thallus. So you have the thalloid structure on rocks, growing on rocks. Okay, these are the male thalli. You can see the kidney-shaped sessile male receptacle. This is the vertical section of the antheridium. Okay, and you can see the archegoniophore is not very clear in this picture. This is the VS of the archegoniophore, the venter with the egg, the neck canal cells, and the cover cells. This is the developing archegoniophore, few thalli which mature archegoniophore or the female receptacle. And you can see the sporophyte, okay, developing in the archegonium with a small seta and sporogenous cells. Okay, there's a capsule, small seta and the foot. Okay, and you see the spores inside the spore capsule. Okay, and stalk is not visible. It is looks sessile. The seta is very very small. Okay, so and you can see the spores tetrads in this picture. Yeah, this is another picture of the Reboulia hemisphaerica with the spore capsules. Yeah, this is another good picture of the organ of Reboulia with archegonium. Okay, with the archegonia. So when you have again vegetative reproduction is only through fragmentation. You don't have any gemma production here. So chiefly they have studied the sexual reproduction as in detail. So sex organs are, as I told you, they are monoecious. The antheridia are, and the archegonia are born on the same thallus. Okay, and antheridia is slightly raised and the archegonial receptacle is lifted up on the stalk during maturity. Okay, and they have seen it is seasonal. The archegonia appear in the autumn, fertilization occurs and the embryo develops immediately. But the sporophyte does not mature until the following spring. So you have a different time gap of around 3 to 4 months between the fertilization and the sporophyte maturity. So that is the introduction of the sex organs. When you talk about the male receptacle, I showed you it's a kidney shaped sessile structure. So it is actually compact in the beginning, but it is deeply embedded in the dorsal notch portion. Okay, so this is the structure of the antheridium, growing antheridium, and it actually the mucilage, you also see mucilage cells. Okay, elongated cells with a distinct nucleus, vacuolated cells developing along with the antheridium. Okay, which are again protective in function, and they must be having some role to play in fertilization. Okay, so the receptacle is raised only to a small, see it is having a very very small stalk. So you can see this is the dorsal portion of the thallus. You can see the male receptacle getting formed on the margins of the thallus. Okay, the stalk is very very small. And sometimes some people have seen that they have seen fungal growth in the near the in the receptacle. The fungal hyphae of the tissue will penetrate the stalk. Okay, so they have seen fungal hyphae in the male receptacle also. So to summarize, the male receptacle occupies a dorsal position, okay, posterior to the female receptacle. That is just behind the female receptacle. So the receptacle is lunate in outline, okay, circular or more or less irregular. So it is sessile. Sometimes it is elevated on a very short stalk. It is monoecious, okay, and the male receptacle represents the elementary stage of branch system. So they say that male receptacle in Reboulia has led to the branching system in the advanced forms of Markantiaceae members. Okay, and you can see here in some cases they have seen the archegonia also developing on the same side as the antheridia. Okay, in some instances they have seen both archegonia and antheridia developing at the same uh, same surface, deeply embedded in the tissue. So antheridia, as I told you, it is sessile lunate receptacles. The receptacle is cushion shaped and raised at the level of the thallus, but the antheridia are deeply sunken into the tissue. That's what is given here. Even though they develop at the surface, the formed antheridia are deeply sunken into the tissue. You see here how they are sunken deeply into the dorsal portion of the thallus. So 4 or 5 antheridia are usually seen in cross section. So some cases, rare cases, you see 24 numbers. Some cases, some strains, they have also seen 16 to over 100 antheridia in a particular, in a single male receptacle. Normally 2 to 3 antheridia are produced on the thallus before the appearance of the archegonial receptacle. So before the appearance of the female receptacle, you see 2 to 3 groups of antheridia are produced. So they are formed close to the apical cell. Okay, so this is actually the place where the antheridia are embedded. Okay, and at the top it is lined by the epidermal cell, which come together to form a structure with a pore in between. So the antheridium is developing here in the inside portion, and the matured sperms get released. They come out through the pores. So you have seen here, you will have the pores here. Okay, the antheridium will release the sperms, and the epidermal cell, the structure is not clear here. So you can see the structure will be like this. Epidermal cells will join together and form in such a way that they form a pore. Okay, and the sperms will escape from the male receptacle or the antheridial tissue through the pore. So this shows the development of the antheridium, how the antheridium is produced. Okay, so here you have just one initial giving rise to the whole antheridium. So this is the one cell which divides mitotically. Okay, and each subsequent cells divide mitotically with a horizontal divisions and giving rise to 4 cells, and then each of these will divide transversely also. You say periclinal division. Okay, and each one will divide transversely to form 2 rows of cells. Okay, and in the end you will find the antheridial portion is differentiated into an outer portion and a middle portion. So a jacket layer and the androcyte. These androcytes are going to give rise to the sperms. They are going to get converted to sperms. Okay, and this is the jacket layer which protects the antheridium, developing androcyte. And this is a small stalk, very, very small stalk. And these are the mucilate cells which are produced along with the antheridial initials. Okay, along with the antheridium, these mucilaginous cells are produced. So that is what is given in the notes. Because the mucilate cells, the main function is to secrete a high amount of mucilage, which will help the sperms to move and get dispersed. So mucilage cells play a role in dispersal of the sperms as well as protection of the immature androcytes. When you talk about the female receptacle, generally in liverworts, you see the female receptacles are produced on the dorsal surface. Okay, and on the dorsal surface like this, on the convex portion of the dorsal surface, and then later on the portion, the surface, they start elongating in this manner, and finally the archegonia lie on the undersurface of the dorsal portion. So this is called the carpocephalum, resembles the head. Okay, cephalum means head. So it resembles the head. So it is otherwise called umbrella. No, this is umbrella shaped structure is called the carpocephalum. So most of the thallus liverworts, they have this kind of umbrella like structures which bear the archegonia. So they are called the carpocephalum or the umbrella top. Okay, so it is a gametophytic tissue, and the upper portion is called the female receptacle. So it is bearing the archegonia. Beginning it will be on the upper surface, and then it will move to the sides because of the elongation of the middle portion of uh, the receptacle. And on further elongation, the archegonia are pushed to the underside of the receptacle. So they will be as though they are inverted in an inverted position. The receptacle is initially on the thallus surface, and then the stalk grows to raise the thallus, and so the receptacles are pushed, archegonia are pushed sideways. Finally, they are pushed on the undersurface. So that is how they come to lie in the undersurface. So this diagram shows the seta, okay, and the sporogonium, the carpocephalum with a mature capsule. Okay, and here you can see the capsule is, the capsule wall is broken, and the spores are shown outside, ruptured. The capsule wall is ruptured, and you can see a mixture of spores and elaters. You can see the mixture of spores and elaters. When you talk about the archegonia or the female receptacle, again they are dome shaped structures which I had shown you. This is how they are formed. Formation of the archegonial receptacle involves the apical cell of the thallus. The same apical cell which favors the growth of vegetative growth of the thallus also forms the archegonial initial, you call it, archegonial initial. So wherever you have the apical cells, you find the development of the archegonial initial during the development of the sex organs. And the initial arises as a papillate cell, okay, as a projection, and then you find vertical divisions on the outer cell and the inner cell. You have the basal cell and the apical cell. You have the basal cell and the apical cell. The basal cell will go on to form the venter canal cells and the venter. Okay, and this is how it matures. The venter canal cells and egg canal cells are formed by the uh, basal cell. This is venter, which bears the egg, and it is lined by the venter canal cells, and you have a long neck, nearly 6 to 8 celled, which are again lined by the venter canal, neck canal cells with the cover cell at the top. So archegonial stalk and the neck, they curve upward, okay, and outward, so as to facilitate the entrance of the sperm. So mature archegonium contains 18 to 20 neck canal cell which break down soon after their formation. So you'll have mucilaginous covering. Here also you can see mucilaginous cells, and in a great majority of cases only one archegonium is formed from a single segment of each apical cell, and only the one egg develops into a sporophyte. Even though there are 4 to 5 archegonial initials or the female receptacles produced, only one is matured, and slender filaments arise below each archegonium and grow as and normally they have the protective function. The filaments become quite numerous in each receptacle notch, persist till the spores are shed. These are the filaments I had shown you. These are the filaments. Okay. Another picture, it was quite clear. You can see the filaments very clearly in this picture. Okay, so they are also produced on the long side of the archegonium. So as the development of archegonium proceeds, filaments are also produced, and they are protective in function. And the mature egg is oval and contains plastids and several large oil globules, probably reserve food material for the growing embryo, because there is a 4-month gap between the embryo formation and development of the sporophyte. So that is why the egg is rich in reserve food materials. And the mature egg does not develop a cellulose wall until after fertilization. So the number of lobes developed by the mature female receptacle corresponds with the number of growing points organized in the young receptacles. So fertilization, the male and the female are quite close because they develop the male receptacle develops just behind the female receptacle.